Welcome back everyone to the Annual Behavior Live Annual Online Conference 2024. Now we'll start off with the first workshop. The workshop is on how to build your own insect detecting AI camera trap. We have with us Maximilian Sittinger, who is from the Helmholtz Center for Environmental Research, and Michael Watson, who is the CEO at BeFriendly, and they'll be talking about the insect detect system that they developed.
Okay, I'm going to share my screen. Let's hope that it works. So now you should be able to see the presentation in full screen. Is this okay?
Yes, this looks perfect.
Okay. So thanks for the introduction. So you already know that I'm coming more from a science background, and Michael is like the guy who puts our ideas more into reality, and he will have the second part of this talk or this workshop, and we'll give you more information about how they implement the system in their smart bee hotels. So okay, so this works. So I just want to give a very short introduction why we actually need insects, because it's not only that they are beautiful animals and we like them a lot, we are also actually very dependent on insects. So for example, many insects are important for nutrient cycling, for pest control, and especially pollination is very important. So if you like peaches or other fruits, you should also like insects, because actually most fruits are dependent on pollination by insects. So otherwise, you would not be able to eat peaches or apples without them. It's estimated that pollination alone, it's between $235 to $577 billion per year to global agricultural productivity. So this is something that's actually also from a financial point of view extremely important for us. And on the right, you can see a plot where you can actually see that many different crops are very dependent on pollination. So if we wouldn't have pollination by insects, we would lose a big part of agricultural productivity, actually. You might have heard this in the media since a few years, that insect abundance and diversity is declining. So in this scope of insects being very important for different ecosystems, ecosystem service for us is a little bit alarming, because we could expect in the future that if this decline is going on more strongly, we will also lose these ecosystem services, actually. So what you hear in the media, there's often this phrasing of insect apocalypse. This is a little bit simplified, so actually it's a little bit more complicated. We actually see different trends depending on the system where you look at, depending on the country. So decline is not always decline. It's a little bit more complicated in reality. We already know most of the drivers that push this insect decline further, but it's very difficult to actually differentiate these drivers and the effects between these drivers, because most of them are correlated. And if we want to design more effective conservation strategies, we actually need more monitoring data. So we need data on insect population changes with the highest possible resolution to be able to differentiate these drivers and calculate the exact effects, to be able to find solutions on how to stop this decline of insects. In the project Monvia that is running in Germany since a few years, we are working on concepts for nationwide insect monitoring. Our main objective in this part of the project was to find potential indicator species, for example, hoverflies or predatory bugs, that we can monitor in agricultural landscapes to get information about the overall insect diversity and abundance, for example. For this sampling, we also developed an automated method, the camera trap that we are talking in the following about. So this is just a little bit of background where this camera trap actually comes from. But then another important question is, why do we actually need automated insect monitoring? Because we already have traditional methods. For example, on the bottom left, you can see malaise traps. These are these tents. You can see pen traps with different colors that attract, for example, pollinators, and sweep netting is also like probably the most traditional method to collect insects. And these traditional methods give us, or can give us, a very high taxonomic resolution, so you can identify the species. They're somewhat standardized. For example, for pen traps, many people use different colors, and the color is actually very important for the insect attraction. So I wouldn't say they are like completely 100% standardized, but they're definitely more standardized than the automated methods. For automated methods, at the moment, we still have to expect a much lower taxonomic resolution. For example, from insect images, you can only go to a specific taxonomic level. Many insect species are actually not able to be distinguished from images alone, because you would have to look at very small hairs on their legs to differentiate species. And at least at the moment, because there's still a lot of development going on, these methods are not yet standardized at all. Now, to the problems for traditional methods. Traditional methods take a lot of time. It's not only putting the trap out in the field and collecting the insects. What takes actually a lot more time is identifying the insects under most of the time under a microscope, one by one. And this, of course, is also expensive, because you have to pay a taxonomic expert to identify these insect samples. So we often only have snapshot data available at low spatial temporal resolutions. So for example, in most monitoring projects, we would go in the field 2 or 3 times per season, have a collection for 1 day, maybe for 2 days, 48 hours, and that's it. So we only see a snapshot of what is actually going on in the field. Another problem is that for most of these methods, the insect have to be killed, which is not nice, of course. For the automated methods, they can be much more cost efficient, because you put a automated trap outside in the field. It collects the data autonomously by itself. And because we implement some AI classification pipelines, we already have the process data available immediately without any expert having to look through all of these samples. And the insects are not killed, which is nice, because you can deploy this camera trap for a whole season every day without having to worry that you're killing lots of insects. So our work is built on work in a whole ecosystem, or like a small scene of automated monitoring researchers. So I just wanted to give you a few papers that were, for me, very important to actually go down this path. And for anybody who's interested more, there are really cool groups out there who are just growing at the moment. And everything is still very flexible and dynamic in this scene. So now a little bit of information to the insect camera trap that we have published this year. So this is basically an artificial flower platform that is an insect attractant. So similar to pen traps, where you have different colors that attract insects, we use this artificial flower platform. There are also many different designs possible. So this is just what we ended up with. On the right image, you can see a little bit of the electrics intrinsics of the camera trap. I will give a little bit more details on the next slides. And this whole build, so from the hardware assembly to the software setup and so on, is completely published on our documentation website. So this is also the idea behind making it accessible, that people can actually use our developed camera trap for their own projects in private, for their garden. So it's not that difficult to build this yourself, actually. So on this documentation website, you will find links to, for example, the GitHub repositories where we published all the codes. We have published all the models that run on camera trap, so the detection models and also the classification models. You can find a list of components that you have to buy to build this camera trap, and step-by-step instructions on how to assemble everything. At the moment, we only have the current version published there, but I plan later this year to also published the new version, which I will show you on the next slides. So for the current project that we are working on, we are interested in plant-pollinator interactions. So for plant-pollinator interactions, you need the real flower as background, because you want to basically find, for example, network structures, which insect species visit which flower species. So we cannot use the artificial flower platform for this. To be able to put a camera on top of a flower, we basically split up the design that the camera has its own enclosure and the rest of the electronics is in another enclosure, that you don't have like a big box hanging on top of a flower. We have 2 batteries connected to the system. At the moment, there's not yet a solar panel, but we plan to implement a solar panel for long-term deployment also. Everything is integrated in waterproof enclosures. This time we have a button on the outside, so you actually don't have to open the enclosure in the field. You can just press the button and the camera will turn on. And the cost is about 500 euros, but depending on the kind of batteries that you use, for example, if you add a solar panel or not, I would say the cost is somewhere between 450 and 650 euros, depending on the quality of the components, for example, that you use. Is there a question in the chat up to now, or should I just continue?
Sorry, I think you can continue. I don't see anything
Okay.
coming in as of yet.
Okay, yep. So the main reason we have this new design is to monitor flowers, but we are still using a platform, but we simplified the platform a little bit to resemble the pen trap station that we also use in this project. So we have a yellow, blue, and white circle basically on this platform, and this are exactly the same colors that we also use for the pen traps to be able to compare the catch or the insect species that we get with both methods. So this camera trap is built on 3 main components. Probably the most important component is this ORC-1 camera or OpenCV AI kit. This 12 megapixel camera that's directly connected to an Intel chip. This is like an AI capable chip where you can run on-device object detection and tracking. So this is what makes the camera possible to use AI with in real time. For this camera, we need a host. So for the host, we use the Raspberry Pi Zero 2W, which is like a mini computer that's easy to program and nice to work with. And for power management, in the published version, we use the PyJuice Zero power management board. Now for the new version, we use a different power management board. So there are several options available. This is a little bit more cheaper. This is the main reason why I use it. And power management is basically needed to implement like a schedule. So when to turn on the system, when to turn it off, and then to have some like scheduled recording times. There are also already different variations from different people. So Michael will tell you more about the Smart Bee Hotel and their modifications. There are also people who use this camera for dung beetle monitoring. For example, here in the middle bottom, you can see this piece of cloth. There's this basically on top of a poop pile. So they use poop to attract dung beetles, which is really funny. And it works. It actually works well. You also have an implementation for ant monitoring. So this is this bait station on the right side. So you can use this camera trap not only for pollinators, but also for different kinds of insects, basically. To be able to detect the insects, we first needed to train a detection model. So for the detection model training, this dataset is also published at RoboFlow, for example, or at Zenodo. We annotated about 1,300 images with insects sitting on the artificial flower platform with a bounding box. So these blue boxes that you can see here, this is basically the information that you have to give the model to train it to detect these insects also later on live stream frames. We have also published a Google Colab notebook. So Google Colab is basically a platform where you can use a GPU or graphics card for free. This is what you need to actually train the AI models. So you can use this Colab notebook without having to go deeper into the code to train a model on your own data set. Maybe you're interested in monitoring dung beetles, for example, and you have collected images of dung beetles, annotated them, and then you could use this notebook to train your own custom model, basically. So we use the YOLO architecture. YOLO stands for you only look once. This is basically one of the fastest and most compute efficient model architectures that are published at the moment. Now it goes until YOLO 11, so every few months a new version is coming out. But by default, we still use YOLO v5 because we have made the best experience with it. I will not go much more into details about the model metrics, but this is basically telling us that most of the insects in our test data set are detected after model training. To be able to run this detection model on the camera itself, you have to go through several conversion steps, but this can be done with an interactive web platform that makes it really easy. So you basically only upload your model weights file, and then the conversion is happening in the background without having to write any code or going into the details. So for this year, we actually want to monitor insects visiting real flowers, but we only had a model that was trained on this artificial flower platform, so like on a homogeneous background. And for heterogeneous background or complex background, like different kinds of vegetation, the challenge is actually much more bigger. So it's much more difficult to train a model that works with this more difficult background. But fortunately, I'm now, so since this year, I'm working together with Valentin Stefan, who already collected images of insects visiting flowers with smartphones and annotated these images to train a YOLO model. So we already had the dataset basically available and adapted this dataset from Valentin to train a model that can now run on the camera itself. So in the right image, this is not a real insect. Oh, it is a real insect, but it's like a dead bumblebee. This is just for to showing how the model works in real time. So the insect basically visits the flower, gets detected, and from this time on, we can trigger the image capture that no images are taking when there's no activity, which is true for most of the time. These models can actually be very easily switched out. So these are only 2 lines in the Python code that we use. The rest of the model information gets extracted automatically from a config file that you generate after converting the model. So you can easily use different detection models depending on your use case. So if we only run a object detection model, we get information about the insect for each frame. So if we run the camera at a frame rate of, for example, 20 frames per second, we would get 20 insect detections per second. But we are also interested in having a kind of abundance information. So we actually need to distinguish individuals, and to be able to distinguish individuals, we need to implement an object tracker. So this object tracker basically follows the insect detection between each frame and assigns a unique ID to each individual. So with the object tracker implemented, we can avoid multiple counting of the same insect as long as it is present in the frame. If the insect moves out of the frame and comes back in again, it will be counted again. So this is something that we cannot avoid at the moment. But this is also similar for some traditional methods. For example, if you do transect walks, there's also a high or relatively high probability that you would count some insects, the same individual, multiple times if it moves around. So with this object tracker, we can also get an abundance estimation by distinguishing individuals per frame. So I won't go too much into detail for the whole processing pipeline that is running on the camera itself, but basically we had one big problem that we solved with this pipeline. We need a detection model, so this YOLO model, to run as fast as possible for the object tracker to work as accurate as possible. Because if you have a high frame rate, the distance for an insect that is moving in your frame will be smaller from frame to frame compared to a low frame rate. So for example, if you only have 1 frame per second, the distance that the insect travels in this time would be larger, which means it is harder for the object tracker to keep the track stable. So the faster the model can run, the more accurate our object tracker will be. To be able to run a model fast on this camera, we need a small image resolution because the model always looks at each pixel. So the less pixels you have in your image or in your frame, the faster the model will run. But if we downscale the image to, for example, 320 by 320 pixel, the insects will get really pixely. So we will have a low resolution, and then it will be more difficult to classify the insects in the next step. For this reason, we actually run 2 streams in parallel, one low quality stream and one high quality stream. So the low quality stream is only used as input for the detection model and for the object tracker. And then we synchronize the detection. So these are basically the red bounding boxes with the metadata that we get from the tracker with the high quality frames. And this is what we save on the Raspberry Pi's SD card. So this is basically the crop detection that I will show you on the next slide. And this is what you can then work on in the next steps, for example, to the classification. So on the left, you can see, on the right, you can see an example of this low quality frame. This is the input in the detection model. So the insects get detected on this frame, and then we synchronize the bounding box coordinates with the high quality frame that you can see on the left, and then crop the detection. So these are basically, this is the area inside of the bounding box. And these insect images you can then use in the next step for classification. For the classification model, we also published a notebook. So this is basically independent of your detection model. There are multiple reasons why we split this up into 2 different models. For example, it's easier to train a classification only model because you don't need to draw bounding boxes. So you basically only take the insect images, put them in individual folder. Each folder would be your insect class, for example, species or family. And then you use this data set to train one of the different models that are supported by this YOLO v5 training. We ended up with a fishnet because this got the best results for us. Then you can also, inside of this notebook, you can export it to Onyx format. This runs faster on a normal CPU. So to train a model, you would need a graphics card, for example, in form of this free Google Colab GPU instance. But to run a model during inference, so inference means you run a model over your captured images, you don't need any special hardware. So you can actually do this on your complete normal standard CPU laptop, that's no problem. For the classification model that we have published, we have 27 classes. And as you can see, for some groups, for example, for hoverflies, we can go down to species level for some common species that are actually relatively easy to distinguish because of their species-specific patterns on the back. But then for other species, for example, for wild bees, we only have 1 class at the moment. So this is a very a coarse resolution model at the moment. And we're actively working on making this bigger and adding more classes to go down to deeper taxonomic levels. As you can see in this confusion matrix, the model is very precise on the test data set. But that said, the test data set is already a part of our curated data set that we selected. So in real world use, this would not be as precise as you can see here. So for almost, for most of the classes on this test data set, we have a very high accuracy. But for real world use, you would expect a little bit more realistic accuracy of maybe 70–80%. if the images were captured in a similar resolution and background as in our dataset. So the last step that we have to do to actually be able to work with the data is to post-process the results that we get from the classification model. Because if we take an image of an insect, for example, every second, this is the default image capture frequency, we end up with multiple images for one tracking ID. So for example, if the insect was present for 10 seconds, we'll have captured 10 images of this one individual. And the classification model doesn't include the tracking ID as information. So each image that is input in the classification model is analyzed separately. So if the insect is moving around, for example, you can end up with different classifications for your sequence that is one tracking ID. And we actually want to merge these different classification results that we have only one row for one tracking ID, which we estimate to be one individual, and then this would be data that we can work on with in the next step. So we also published a post-processing script that first removes all tracking IDs with less than 3 images, because we expect that these are often false tracking IDs, for example, of something that was only shortly or that was flying over the platform very quick. So everything that was tracked less than 3 seconds, we basically removed by default just to have a more conservative estimate. In the next step, this is happening automatically. The classification result with the overall highest probability per tracking ID is calculated. So this is basically a kind of a weighted probability. And then a CSV file is saved in which each row corresponds to one individual tracked insect. So we merge the information for each tracking ID. And this is then data that you can work on later with, for example, in R, in your standard analysis pipelines. There are also some overview plots created, but this is just to get a rough feeling of the data. I would always recommend to create your own plots later on with your statistical software that you use. So I'm going to rush a little bit over these slides. So basically, I just want to show some examples that we also have published in our paper from our pilot test last year, where we had 5 camera traps deployed at 5 different orchard meadows with a total recording time of 1,919 hours. We ended up with about 25,600 unique tracking IDs after post-processing, of which about 8,700 were no insects. So basically these were, for example, bird poop. This is what we have on many images. When a bird poops on a platform, has a high chance that it gets detected as insect, and we end up with a lot of images of bird poop. We also can see leaves on the images, leaves falling on the platform. We have on class for insect shadows. So a shadow would also be detected and then filtered out during classification. Yeah, so not all of these images were insects. If you look at the plot on the bottom right, chemtrails merged. This is basically the merge data from all 5 camera traps, and here you can see that flies and small flies were the most abundant classes, and this is what you can also see on data taken with traditional methods. The standard flies are probably the most abundant group that you can see under normal field conditions. But for us, what was nice to see that we also actually captured a lot of hoverflies. And hoverflies are super interesting for our work and our research at the moment. So we could actually go a little bit more deeper into hoverfly activity because we have this very high temporal resolution. We could plot the hoverfly activity per day for the whole season in 2023. And we can actually see some interesting behavior, for example, peaks in end of June, beginning of July. And this corresponds to the data that we also got from traditional methods. So we can actually see activity peaks from which we highly expect that they are really happening in reality. So this is not only a snapshot that we get from the traditional method, but something that we can actually see for a whole season. Okay, we found the highest activity end of June, and we are pretty certain that this is really the case. We could also look into daily activity. So this is interesting, for example, for hoverflies, we know that they're more active in the morning hours where it's not so hot and more nectar is still available in the flowers. This would be the opposite for bees. So bees are known to be a little bit more active in the afternoon hours, for example, as comparison. So in the chemtrap data, we could see this pattern that we already know from the literature or from traditional methods, that the hoverflies that we captured with the camera traps were actually more active in the early hours of the day compared to the later hours. So just a little bit of information for the new project that we are working on since this year. So this is called the SEPI project, Standardized European Monitoring of Plant Pollinator Interactions. And we are mostly interested in interactions. So which species interact, which pollinator species interact with which plant species. And we also have some applied question that we are asking, for example, which pollinator species are the most important? So which one provide the most pollination services? This project is happening in 8 countries. So since June of this year, we have 58 camera traps deployed in these countries, and we compare traditional methods to the automated camera trap. And basically, one of the goals of this project is to find spatial trends in pollinator diversity, composition, visitation rate, and network structure. And if we end up finding similar trends with the automated method compared to the traditional methods, we would basically be able to use the the automated methods for the coming years and know that the information that we get from these automated methods corresponds to the information that we would also get with the traditional methods. This is more or less the goal. For this validation, we are doing transect walks, so basically walking through the site and collecting insects with a sweep net, and also count pollinators that visit each flower. On the same side, we deployed the camera traps on top of flowers to be able to compare the data to the transect walks. But we also have pen trap stations, so this is like a blue, yellow, and white bowl, where you collect insects and compare these to the camera traps on top of a platform. So you could see this image in one of the slides at the beginning, where you have these platforms with the 3 different colors that correspond to the pen trap colors. Here are some examples of the first images that we got from this year. So you can see, depending on the insect species, for example, for butterflies, we can actually go to species level for the classification. For some hoverflies, it's also possible. But then, of course, you will always have some flies or bees where you can only go to group level. So, for example, to family or to genus. At the moment, I'm working on an interactive web app to make it easier to communicate with the camera trap in the field. So this will enable users to actually connect their phone to the Raspberry Pi, to the camera, and then see the live stream and do all of the configurations over their phone. Because at the moment, you still have to log in via the terminal and SSH, and this is already a big hurdle for many people. So this web app will actually make it a lot easier to play around with the camera in the field. So that's it from my side. This was basically some background information. Are there any questions yet in the chat, or should we move on?
I think we have one question. Maybe we can just time before you move on. So it's from Catherine, and she's asking if you have ever tried to place one of these setups in a more urbanized location, and they want to know if some species of insects are detected more abundantly in these urban areas.
So we haven't done this yet. I think there are probably some people who are doing this at the moment, and maybe Michael can say something about this, because they're actually deploying some of their camera traps in urban environments. And yes, I would expect a different species composition in urban environments compared to like field environments or agricultural environments. Does this answer the question, or was there still something that?
I think for now, yes. Yeah. Maybe I ask one quick question before we move on to the next section. I think I was interested in the object tracker that you mentioned. So the videos that you showed had the insect moving over the flower, but I guess in some cases with the depending on the flower and the insect species visiting it, they're likely to go on the side or around and stuff. And so this is obviously going to create occlusions, right? So how does the object tracker deal with it? Are you able to still get the same identity back, or?
That's a very good question. And occlusions are actually the biggest challenge that we have regarding the object tracker. So the object tracker uses the information from the detection model. So the more precise your detection model is handling occlusions, for example, the more precise also your object tracker will be. So for the data set that we are working on at the moment, we actively search for images where only a part of the insect is visible. For example, if the insect moves inside of the flower and you only see the bud or the behind of the insect, we use these images especially to annotate them for the model training. Then the object detection model will get more robust against these scenarios, and the object tracker will also be more robust towards occlusions, for example. So we basically try to make the detection model as accurate as possible, which will help the object tracker to deal with occlusions. And so from my experience, about 1 or 2 seconds of occlusion is still okay. So even if the insect is completely lost during this time, the same ID is assigned if it pops up again after 1 or 2 seconds.
Wait, thank you. Catherine says that answers her question. So maybe we can move on to the next part and then wait for more questions in the chat.
Okay, then I will give over to Michael.
Yes, thank you. Thank you, Max. So in the next couple of minutes, I'd like to show you how we have used Max's system, and we've made some small modifications on hardware, but also in software to adapt it to our use case. And with this presentation, I just also want to show how easy it is to modify InsectDetect system to your particular needs. So just real quick about who I am. My name is Michael Watson, and I founded the startup Be Friendly 3 or 4 years ago. And what we do is we implement evidence-based conservation measures, and as the name suggests, with a focus on native wild bees. So our focus is on creating habitats for native wild bees. And we also want to show the world that nature conservation is fun. So it's not always restrictive. We also want to show that it can be fun and creative. And yeah, hope that we do that well. So we build an artificial nesting habitats, also called bee hotels for wild bee species in Germany or Central Europe. We also implement, conservation measures in the outside. So we sow wildflower meadows. We create habitats for wild bees in all sorts of ways. Aside from these implementations and real conservation measures, we also host a lot of workshops. So we do that with kids, but also with teenagers and grown-ups. We have a nonprofit and a for-profit branch for Be Friendly. Nonprofit, obviously, for schools and for other educational institutions, and for-profit then for corporate events. So we do like team building events for big companies, in which we show employees how to build artificial nesting habitats for wild bees that actually work. Because a lot of times, a lot of bee hotels that you can buy in stores just aren't made very well. So yeah, we really try to stick to the evidence-based conclusions from science, and yeah, work with people like Max and all kinds of other scientists to just be at the forefront of current developments. Yeah. So our goals with insect monitoring is, for one, we want to monitor the effectiveness of conservation actions. So we, like I just said, we implement conservation actions. We try to create habitats that actually work for to attract wild bees. We know that our actions and that our measures are good and that they actually work, but of course no one else knows that. So we want to really provide data on how effective these conservation measures are. Our second goal is to make the world's first smart bee hotel, which is kind of a marketing gag, obviously, but yeah, we really want to show that our measures, or also other measures that are implemented, yeah, we want to measure the effectiveness. And then our third goal with insect monitoring is to provide CSRD compliant biodiversity data. CSRD is a European regulatory initiative. It's short for Corporate Sustainability Reporting Directive. Next, Max, please. So I'll try not to bore you, and I'll try to keep this short, because as often with regulatory themes, it can be a bit dry and boring. But CSRD is the initiative by the European Union to have companies and other big institutions report on their sustainability, their impact on sustainability throughout the year. So that includes climate, pollution, water, also sustainability in the sense how our employees treated, and how our how do companies engage with local communities. But also one big chapter of CSRD, so of this reporting directive, is biodiversity and ecosystems. That's what we focus on. That's what where our expertise lies. And biodiversity and ecosystems, this chapter of CSRD, is subdivided into 123 data points, which try to monitor and to collect conclusive data on the state of ecosystems, the state of ecosystem services, and also the state of species. In the context of CSRD, there are several species that are especially interesting. For example, how many threatened species are there near a company location? How many invasive species? How many pollinators? How many pests are at any given location? So to do this, we had to modify Max's system slightly, which we I think we did in a constant ping-ponging emails back and forth, and in collaboration with Max. So thanks again for all the help with that. The first steps we did, the first modification steps we undertook, was to build a wood enclosure, and also add a nesting space, so a bee hotel, to the monitoring system. So here we use local wood. It's Douglas pine, which is you can use outside, and it can stay outside for up to 15 years without having to be treated. So it's one of Germany's most more durable woods that we can get from here. We had to weatherproof the whole system, because the goal is for it to stay outside all year, or rather between March and October, when insects are actually out. We 3D printed a case for the insides. Yes, thank you, Max. And yeah, but more or less we have a very similar setup. So here in the picture, you can see the green case inside. That's where all the hardware is. So the Raspberry Pi, the Oak camera, and then in the bottom hexagon, you can see the landing platform. Here in this picture, we experimented with fluorescent blue because that is the color that is most attractive to native wild bee species in Central Europe. But we also tested all kinds of other colors, fluorescent yellow, also with some stickers, with flower stickers on the landing platform. So yeah, once we've processed the data of this here, we'll know a lot more about what worked best. Then another thing we tested this year was how does it compare if we use solar as a power source, which obviously you are limited to your recording intervals can be shorter because you just don't have enough power if the sun isn't out for a couple of weeks, and also tested how well it would work if we have a constant electricity, so hooked up to a cable. And yeah, so those are the main modifications that we tested this year. We had several different test environments, and also here we worked with a lot of research institutes. For example, the University of Applied Sciences in Bingen, where they have a study going on evaluating resource-saving, cooling, semi-intensive green roofs on water, energy, and biodiversity. So they have a lot of climate data and temperature data, but before this year, they had to measure biodiversity, especially for insects. They had to have students come and trap insects with the more traditional techniques. But as of this year, they were able to use our monitoring system to collect data on insects, which for us obviously is also super interesting to then compare at the end of the season, how did our data hold up compared to the more traditional techniques. So another very interesting study we were able to participate in is in the German Research Institute for Horticulture in Heidelberg, where they compare the attractiveness between native plants and related cultivars. Here we tested our monitoring system without the solar cell, with the cable, so a constant electrical connection. And at first we were worried a bit that it might cause overheating, especially in summertime when it gets quite warm here in the south of Germany, so it can easily be 35 degrees. And then with the camera, the Raspberry Pi running all day, it can, we were a bit worried at first that it wouldn't hold up to the temperatures, but yeah, so now we know that it didn't cause any errors, and it seemed to work fine. Aside from these very scientific and research based projects that we're involved with, is we also want to use the monitoring system as an educational tool for all ages. So more for children and teenagers, but also for grown-ups. And we have a few monitoring systems at natural history museums in the Palatinate here in our area, and also in the ecological gardens in Mannheim. That's where I live. And yeah, we want to use the data that we generate for STEM workshops for children, in which we show the pros and cons of automated insect monitoring compared to more traditional methods. And also, of course, to collect insect data on museum grounds, which is interesting for natural history museums. Oh, and maybe just if you could go back one sec. Here in this picture, we were actually running some tests on collecting data on insect-specific insects. So here we have we were collecting data on mason bees, Osmia bicornis, and yeah, so we had them in this lab setting for a few hours trying to collect data on them. Our biggest project to date is together with the Fraunhofer Society in Stuttgart, Germany. Here you can see a green wall, which is not just a green wall, because a lot of green walls currently have their main goal is to help with climate-related factors. This green wall is specifically designed to promote biodiversity by choosing location-specific plants that are supposed to help endangered insect species in exactly this area. So for this, we had to change our wood enclosure a bit as to just mount it into the system that already exists. Max, could you do the next? Yeah, exactly. So then here we kind of adapted our design, our frame design, so that we can just hang it into the modules that already existed in the green wall. Aside from all the hardware modifications, we also had to, or we're also modifying the software. For us, for our use case, not all recorded data are relevant. So our focus, like I just said, our goal is to provide data, CSRD relevant data. So we really focus on abundance, diversity, and information on these species. So invasive species, threatened species, pollinators, pests. Those are the really important classes for us. We're also working with other open source projects and other research projects worldwide as to expand our data set. So yeah, we can use, we can have even more granular results concerning wild bees, for example. So right now, the InsectDetect data set can distinguish between honeybees and wild bees and bumblebees, I think. And we want to, of course, go deeper and then also distinguish between different kinds of wild bees, native wild bees. Another big project for this year is the IoT connectivity via narrowband. So we also want to be connected to our systems all year round, so that we don't have to go out and service or take the pictures from our from the monitoring systems, but be able to access it via IoT connection. Okay, so we also have a preliminary sample report that we did just to allow some time for questions at the end of the presentation. I'd suggest that we skip over these slides, and if anyone is super interested, just give Max or me an email, and then we can talk about it after the presentation. Yeah, so next year, our next steps is our pilot phase with 20 insectors. That is what we are calling our monitoring system, the insector. Testing different contexts, so green walls, green roofs, agricultural landscapes, and also urban landscapes. And also try to solve some methodological questions that we ran into this year. So how many insectors do we need for X square meters? Where do we place them? And yeah, so we want to really be at the forefront of setting standards for automated insect monitoring. Thank you.
Thank you so much. I think we have one question already in the chat. Natasha asked if you managed to get osmia nesting and foraging in your flight tents. And if so, they would be very interested in getting in touch with you because they've been trying to get them to nest in the flight tents, but have not been successful so far.
Yeah, we did.
Okay, then
Yeah.
Natasha, you can get in touch with them.
Yeah, definitely.
I think while we wait for more questions in the chat, we have some time, so maybe I can ask a few of mine, and I think David also has one. So I ask one, and then we get to David. So I saw that you're using the Pi Zero for the as the base for the system. And I think I recently read that they have new models out with an AI kit and stuff like the Pi 5. I'm wondering if you plan to integrate these. I guess they might be better with the problem of how fast you can do inference on the chip itself, right?
So yes, we are already working with the Raspberry Pi 5 and the AI kit, and just recently, I think 2 or 3 weeks ago, the Raspberry Pi AI camera also came out. So this is also super interesting for us. I would say for the Raspberry Pi 5 and the AI kit is a little bit of overkill for what we actually want to do, because it can do a lot. I mean, it's really capable, but it also consumes a lot more power compared to our solution at the moment. So at the moment we need about 4.4 watts of power, and the Raspberry Pi 5 with the AI kit goes more into the direction of 9 or 10 watts. So more than double the amount of power needed. And this is a really big problem for field deployments where you only have a solar panel. So, but regarding the Raspberry Pi AI camera, which is much more resource efficient, I think this is a really interesting tool that we are working on actively to support it with our software pipeline. Yeah.
David, do you want to go?
Yeah, I was going to ask Michael, the Insector product that you're doing, what is the cost of that? And when companies buy it, do they get can they use it to demonstrate their commitment to renewables and so on, and get any tax breaks and so on it?
So for next year, for the pilot phase, because it's still a work in progress, we're planning to sell the Insector module for 3,000 to 4,000 euros. We still have to do a bit of calculating on that, and then also have a monthly fee depending on what how often companies want their reports. So
what's the incentive for the company to do that? Is there a sort of initiative that they're part of?
So for big companies, big corporations have to report on sustainability, also on biodiversity as of 2023. So last year. So they're still writing their reports and handing them in. Small and medium-sized enterprises will have to report, and it's mandatory to report as of 2026, or you can apply for an extension to report by 2028. So this reporting directive, the Corporate Sustainability Reporting Directive, actually mandates companies to provide data on biodiversity, which we are planning to provide with our Insector reports.
So the Insector is an easy way for them to basically promote.
Yeah, exactly.
Activity around their sites, but would they have to also provide food and sort of wildflower areas and so on to encourage the things? So it's all part of one package.
What we want to try to do is to measure the effectiveness of conservation measures, especially if we think about the green wall, which is super expensive. It can cost between 10 to 20,000 euros per square meter. And if you spend that much money on a conservation action, you really want it to work. And to measure the effectiveness of these expensive or also less expensive conservation measures, we want to help provide the data. And if we indeed don't measure a positive impact on insect populations, then we also want to advise and help optimize the green wall system, for example. Maybe change the plants, maybe change the intervals when gardeners come to mow or to cut the plants, and to really create specific recommendations for that very location.
So the companies get measured on how well they're performing. So it's not just they have to report, they have to actually make a difference.
Not yet. Today, companies just actually have to report. There's no sanctions if you don't do well on sustainability. But already today, banks are advised by the European Union to only, or to give out loans at give out cheaper loans to companies that actually achieve their sustainability goals. So there is definitely a financial incentive to do well for companies on biodiversity, but also on climate and pollution. And we want to provide in this report afterwards that we generate every couple of months. We want to provide help and CSRD, the interpretation of the insect data for the regulatory for the reports at the end of the year.
Okay. Do I have time for another question, Avi?
Yes, I think maybe I'm still waiting for.
But before we were online, we were talking about, and you were talking about a project that you're working on with schools in Germany, and that next year there's a science initiative. Could you talk about that a bit and just describe what it is that's going on in order to encourage kids to get involved in science projects? Sure.
So next year, in the context of a Germany-wide event called Explore Science, we want to show school kids how AI or how automated insect monitoring is used, what use cases there are, and also how it compares to more traditional methods. So we want to have kids and teenagers then make up their own minds which techniques they think are better for the future, and show the pros and cons of automated insect monitoring as compared to more traditional methods. Because I think in the end, automated insect monitoring is just going to become another tool in the toolbox of entomologists. It's not going to replace entomologists, and I think our hope for conclusion for kids is then to realize that it's a cool tool to generate data over long time periods, but it's not gonna replace entomologists.
Maybe I can quickly follow up on that, because I guess I mean the way I see this as being different from traditional methods is, in one, you have to say actively go around and catch these insects and try and identify them, and in the case of these automated systems, you're sort of it's a passive process. Like, you have a setup in one place and you're expecting the insects to come, and that's the only way that you can detect them. I'm wondering if there is, I guess maybe I have two questions related to it. One is there a difference in a flower with a camera on top in terms of how much it gets visited? I guess maybe, I don't know, could it interfere with some insects when they try to land? They see this big object looming over the flower, and then they decide not to. And the second question, slightly related to that, is I guess there are some species which are less mobile, so they tend to maybe not move around so much. And then maybe there's a bias in how much these species get detected compared to ones which are more active. Is there something that you looked at in these terms?
So regarding your first question, we definitely expect that the camera on top of flower has some kind of effect, and probably also a bias effect that some species might be more deterred than other species. So, but I mean, it's a little bit difficult to actually test this. We need somebody in the field who monitors, like a real human person who looks at the flower all the time, or a camera maybe with a telescope object lens. I don't know. So it's I think for the current project it's a little bit too time intensive to actually test this. But also we are only comparing camera traps with camera traps. So we have this bias already in the methodology itself. And we have the same bias for all sites, basically, and all species. So I think there's something that could be done in the next step to actually validate this with a real human observing the flower. Then regarding your second question, as for all methods, also traditional methods, we definitely have a bias in the taxonomic range of species that visit these platforms or visit the flower. I think our goal should be, in general, also for traditional methods, to lower this bias as much as possible, that we get a very large taxonomic range of species. But this is something that's a little bit bigger than our small project that we are working on at the moment. But we always have this in mind, yes. And this is a very good point, actually. Yeah.
Maybe one last question that I asked to Michael before we close the session. So I guess Matt's talked about multiple other systems that were already there when you decided to start using Insector, right? So what made you choose InsectDetect? Was there something very unique or different about it that you decided that this is the one that I want?
To be honest, it was kind of a coincidence because Max at the time was currently living and working in Heidelberg, which is right where we live. And I stumbled across his article online and emailed him. Actually, I just wanted to interview him for our blog. And because I thought it was super interesting what he was working on. And then we met up and yeah, we got to talking how we could actually use his system for our use cases.
Okay, I guess that's all the time we have for now. So people in watching us live, if you have any more questions, you can ask Mats and Michael on the Discord channels, or you can contact them. Their contact IDs are available online, and then they'll be happy to answer questions for you. Thank you so much for David, you
I
can
just
go
wanted to jump in and thank Max for all the stuff he puts online to build these things. I've built one, and I would recommend to anyone watching. You know, if you're doing it as just a wired system, you can do it for a couple €100, and it's a lot of fun.
Thank you so much, Mats and Michael, for being with us and talking about the system. Yeah.
Thank you.
Yeah, thank you.
Bye-bye.
Right.